Aloha. Today I will talk about ATP synthesis. This is the culminating moment of the catabolic side of metabolism. The synthesis of energy is the ultimate ending of the oxidative degradation of biological molecules. The degradation of all molecules, fats, proteins, carbohydrates, eventually leads to this point. All the electrons, as a result of the oxidation of carbon, have been loaded on the electron carriers, and they have been donated to the electron transfer pathway that is shared between complex 1 and complex 4. Ultimately, all the electrons will be accepted by oxygen, which will be converted to water. Now, obviously, there's more to the story, but we'll get to that. Now, to start, let's do a little bit of a review. How is the energy of oxidation conserved and then used for ATP synthesis? So I pretty much already mentioned that in what I said a moment ago, but the energy has been stored in the form of electrons that are carried either by NADH or FADH2. These electrons are now, they were offloaded to complex 1, or directly to Q, and then to complex 3 and 4, and ultimately to oxygen. In this process, protons are moved from the mitochondrial matrix to the intermembrane space span between the inner and the outer mitochondrial membrane. These protons now, in the form of a gradient between the two mitochondrial membranes, that gradient will then be used by ATP synthase to generate ATP from ADP and PI. So what is the driving force for moving protons back into the mitochondrial matrix? Well, you probably quickly will reply, the proton gradient. And that is true. It is the proton gradient. But if we're talking about specifically what really drives them inside, part of the reason is that concentration difference. But there is also an electrical potential that drives the protons inside. So it's a combined effect between a chemical potential and the electrical potential. We know it as electrochemical potential. And the reason for that is because the membrane site, or the intermembrane space, is more positively charged. All these protons are moved into that space. On the other hand, positive ions are being removed from the matrix into the intermembrane space. So the matrix in general is negative or more negative compared to the intermembrane space. So for that reason, when opportunity is presented, protons will flow in, driven not only by their concentration gradient or difference, but also based on the charge difference between the matrix and the intermembrane space. Now, what would you do to prove that ATP synthesis and the protein gradient or electron transport are coupled? Now, for the next few slides, we will explore how can we actually prove that indeed is the case. The connection between the electron transport from complex 1 to 4, or from complex 2 to 4, and ATP synthesis is the gradient, the proton gradient. So we need to see how that gradient truly connects the two processes, ATP synthesis and electron transport. You might say, well, we already know that. Well, that is true, we already know that. But how was that established? Is there any way that we actually can prove that if we are allowed to do that, or if we can do that? So to start, let's entertain these ideas. What would happen with the electron transport and ATP synthesis if we disrupt or break only the outer membrane, the outer mitochondrial membrane? So what would happen with these two processes if, let's say, we somehow destroy the outer mitochondrial membrane? And that's the only change that we're going to basically do to the mitochondria. What would happen with the electron transport? Well, if you think about it, the chain of events from complexes 1 or 2 to 4 has not been disrupted. As long as we have a substrate that can donate electrons to complex 1, or as long as we have succinate or anything to donate to Q, electrons will flow. Protons will be moved out of the matrix into the whatever space is outside. Now what would happen with ATP synthesis? Well, if we don't have a membrane, the outer membrane, then this intermembrane space will no longer exist. This basically will be the cytoplasm, or basically the buffer if we're doing this in an in vitro environment. There will be no gradient. Assuming that we cannot really generate a gradient because there's no closed space to achieve that generation of this gradient. So if there's no gradient, what would happen with synthesis of ATP? It will stop. Now, what would happen if we break all the mitochondrial membranes, including the inner mitochondrial membranes, but in large enough pieces? And by saying that, I mean that large enough so that we don't disrupt the electron transport complexes. Well, as long as they're not disrupted, and as long as we have a substrate, the electron transport will go on, and protons will be moved across whatever membrane they're bound to. It doesn't really matter whether it's a piece or a large fragment or a small fragment. As long as they're intact together, electron transport will continue as long as we have a substrate and we have oxygen. Now what would happen again with ATP synthesis? If there is no gradient, since we have broken the membranes and the concentration now of protons is the same, there will be no synthesis of ATP. Alright, so let's look at this situation. So we will use some inhibitors to try to prove that the electron transport is coupled with ATP synthesis. So let's do this experiment where we will have mitochondria and we will keep them in a very controlled environment, and we will measure oxygen consumption and we will measure ATP synthesis. Now we will give to the mitochondria the substrate for ATP synthesis. We give ADP and PI. We see no ATP synthesis. It seems that there is some oxygen consumption. This shouldn't happen because there is no really substrate. Perhaps there has been some, there are electrons available that have been donated and that have flown through the system and been donated to oxygen. So for that reason, we may see some consumption of oxygen. Now let's give a substrate. In this case, we'll give substrate to complex 2. We immediately see an increase of oxygen consumption. Does that make sense? Of course it does, because we're donating electrons. The available oxygen in the system will be reduced to water, and of course that means that we will be consuming oxygen. Well, what happens also with ATP? Interestingly, ATP grows as well. So even though we have given substrate to the electron transport chain, ATP synthesis happens. That means that there is a connection between these two processes. We saw that even though here in this case we did give substrate for ATP synthesis, it wasn't happening because something was missing. And what was missing is the electron transport process working well enough, and now we know, to provide the protons, but we haven't really proven yet whether the protons are indeed the linking moment or the linking fragment for these two processes. Okay, so we've added ATP substrates, we added substrate for electron transport, everything is working, but at some point we will stop the electron transfer from cytochrome C to complex 4 by using cyanide. Cyanide will block the electron transport. Naturally, oxygen consumption will stop because there's no more electron flow to oxygen. But interestingly, ATP synthesis stops as well. I would have expected that it would stop a little bit later because there's still some residual proton gradient, but regardless, the point is demonstrated that ATP synthesis stops as well, even though we're not affecting ATP synthesis directly. That means that these two processes, oxygen consumption and ATP synthesis, are connected. Now let's look at the same example, but in a little bit of a different setting. Now we will add the substrate first. We add just succinate, which is a substrate for complex 2. No ATP synthesis, slight oxygen consumption. We'll talk a little bit about that in a moment. But there's no ATP synthesis. Then we add the substrates for ATP synthesis, and suddenly both processes start working. So why would the electron transport pathway begin to work when we're giving a substrate for ATP synthase? We're not affecting the electron transport directly. Again, that says that there is something that connects these two processes. We're stimulating ATP synthesis in the presence of substrate, and both for the electron transport, and both processes begin to work. And without that ATP synthase substrates, both processes do not work. So obviously they are connected. So we can see that oxygen consumption is increased, ATP synthesis is increased, and now we're going to suppress or inhibit ATP synthase with venturicidin or oligomycin. Naturally, ATP synthesis is stopped. We see that the oxygen consumption is also inhibited significantly. There is some residual function or consumption of oxygen, but for the most part is stopped. And the reason for that is, we can explain this a little bit. Well, since now we know that, well, first of all, why did it stop? If we don't synthesize ATP, why would that stop electron transport? Think about it. What is the purpose of electron transport? To push protons across the inner mitochondrial membrane against their concentration gradient. But there is also a cooperation with ATP synthase, because that gradient is released a little bit by ATP synthase that allows protons to flow back in. Now imagine if you stop that flow back to the matrix. What would happen with that gradient? The gradient will increase to a point where basically the electron complex, the electron transport complexes will not be able to push anymore against that pressure. And for that reason, the gradient will increase so much that it will stop the electron transport as well. I mean, we see that it still functions with some residual small amounts, but for the most part has been completely inhibited. All right, at this moment, let's add dinitrophenol, or DNP, which is a weak acid. It is permeable to the mitochondria. We add it, and suddenly we see again an onset of electron transport. ATP is not affected. still stopped. Why? Because it's been inhibited by oligomyosin. There's nothing to, unless you remove the inhibitor, you can't really stimulate it. But why was the electron transport stimulated in this case? Now think about it. What is DMP? It's a weak acid. Think about where would DMP pass through and where would it end? It will pass through the intermembrane space where we have high concentration of protons. This is a weak acid. That means that the acid will be protonated. And then it will pass into the matrix. And what is the environment in the matrix? Basic, because protons have been removed. Now, if this is a weak acid and it's placed in a basic environment, what would the acid do? Donate protons. So in essence, what are we achieving with the addition of DMP? We're bypassing the ATP synthase function to allow protons to come in. We're using basically another mechanism to channel protons back into the matrix, thus relieving that pressure of the gradient and transferring these protons back into the matrix. In the sense, the pressure is relieved, we get substrate to be transported, protons to be transported across, and that's why the oxygen consumption resumes after we add DMP. Which proves again, ultimately, that the connecting point or moment or factor between electron transport and ATP synthesis are the protons or the proton gradient. The DMP is known as an uncoupler of these two processes. Why? Because DMP makes the two processes independent of each other. Without that weak amino acid that can channel back protons back into the matrix, these two processes will remain inhibited because there is nothing to relieve that pressure generated in the intermembrane space from the concentration, from the high concentration of the protons in it. So you can see how this was actually proven, or how these experiments demonstrate the connection between the two processes. Here's another proof. I really like going over these figures because they're so logical, and they help you think and make logical connections and see the thought flow that is used to prove the connection between electron transport and ATP synthesis. So suppose now that we isolate mitochondria and we allow them to incubate for a while in pH 9, which is fairly basic, and in fairly high concentration, 100 millimolar of potassium chloride, that eventually will allow equilibrium between potassium and protons across the membranes. All right, and now we quickly exchange the mitochondria into a new environment that now has a pH 7, and it doesn't have any potassium chloride, and also it has valinomycin. Now, valinomycin is an ionophore. Ionophores, if you remember from 41, they can cross the membrane, and they transport, this particular one transports metal ions across the membrane following their concentration gradient. So if we are incubating these mitochondria in an environment now without potassium, what would the ionophore do? They will transport potassium from the matrix outside of it, into the intermembrane space and outside of mitochondria. But what would happen with ATP synthesis if we put now these mitochondria that have been incubated in the basic environment, pH 9, into a pH 7, which is fairly neutral though. But pH 7 offers a lot higher concentration of protons now in an intermembrane space. Because of that concentration gradient or difference between the intermembrane space or outside the matrix and inside the matrix, protons will be allowed to flow through ATP synthase, synthesizing ATP. To help this process, the ionophores will continue to move since there's no electron transport here. Well, at least we haven't stimulated it. But the ionophores will start removing potassium, which are positive, from the matrix outside. That will make even easier for the protons to flow in, because remember again, what is the driving force? Electrochemical potential. If the concentration of positive charges inside the matrix is very high, even if you have a concentration gradient of the protons will not easily flow in, because the positive charge of the proton will be repelled from whatever positive is inside the matrix. But, like I said, again, this is just to prove a principle, that indeed the gradient is the driving force for ATP synthesis. That's just another way to show that. Okay, now let's move along. Alright, so structurally, and you're familiar with this from 481 when we talked about ATPases, the ATP synthase has two domains, F node and F1. F node, and the node is from oligomyosin because it's inhibited by oligomyosin, is transmembrane. F1 is the peripheral part of the protein. Now, that's the reason why this is called F1. I've mentioned that when we talked about ATPases, F-type ATPases in 481, that was the first factor isolated by energy producing factor that was isolated by the mitochondria, and that way it was called F1. Okay, now if we remove the F1, protons will move freely into the matrix or through the membrane. But if we remove A1, what's left cannot synthesize ATP. So what is that telling you about their role? F node is obviously a channel. F1 carries the ATP synthesizing catalytic activity of the enzyme. Alright, so you've learned in this class that the free energy change of ATP hydrolysis is negative 30.5. In reverse term, the free energy change of ATP synthesis is positive 30.5 kilojoules per mole. It's not a favorable process. However, on the surface of ATP synthase, the free energy change of ATP synthesis is about 0. Isn't that interesting? Why is that the case? Why do we report this number while the energy of ATP synthesis at the surface of the enzyme is about 0? So this is what normally the energy graph would look like for ATP synthesis, even though this is not exactly accurate, because obviously the delta G is not exactly matching positive 30.5. We start with the enzyme and substrate, low energy level, and then the product has high energy content. But what happens in ATP synthase is actually very interesting. Now this is ADP and PI. The complex between the enzyme and the two substrates has actually lower energy, and it appears to be that the enzyme stabilizes the two substrates. And then it goes through a small energy hill, and then we have ATP now, which appears to be even slightly lower energetically the complex between enzyme and ATP is slightly lower energetically compared to the complex between enzyme and the substrates. But the change is very small. That's what I'm saying. It's about 0. Very interesting. But look what happens now. This is the ATP in solution. Very large energy to be overcome by the cell in order to release the ATP from the enzyme. Again, why is that? And the reason for that is because the active site has a very high affinity for ATP. Kd is about 10 to the negative 12 molar. That's picomolar, basically. Very high affinity compared to ADP, which is about 10 to the negative 5. And then there's this statement in the textbook: the Kd difference corresponds to about 40 kilojoules per mole of bringing energy, driving the equilibrium toward ATP. And at this point, I would ask you this question: do you have a problem with this statement? If you remember how enzymes work, you should be asking yourself a question: how is this okay to say driving the equilibrium toward ATP? So think back, chapter 6, enzymes 481, Biochem 1. How do enzymes speed up reactions? They affect the transition state. Do enzymes affect reaction equilibria? The answer to that is no, they don't. The equilibrium, reaction equilibria are not affected by the enzymes. Then why does the textbook says that? Driving the equilibrium toward ATP. Now, my personal explanation to this is that we're talking about the reaction that occurs within the processes happening in the enzyme. When we talk about reaction equilibria, we talk about substrates free of the enzymes and products free of the enzymes. In that case, we say that it doesn't affect equilibrium. But obviously, within the steps or the mechanism within the reaction, the enzymes can drive, or it's allowed to say that the enzymes can drive reactions towards one of the products, or towards the products, or towards the substrate. That's the only explanation I can offer. But more importantly, what needs to happen for the tightly bound ATP to be released? Now, this is a very strong complex. How will the enzyme release that product? We say 10 to the negative 12. This is if you're familiar a little bit with dissociation constants, this is an incredibly strong interaction. It's almost as if it is covalent. That's how strong it is. So how will that enzyme release ATP? What needs to happen? Well, if you think logically, you would say, well, something needs to make that enzyme lose its affinity for ATP. And that's exactly what happens in this case. We need to force the protein to lose, or I shouldn't say we, but the cell needs to force that protein, or ATP synthase, to lose its affinity for the ATP so that the ATP will be ejected or released. Now, that's where the protons come at play, and that's how they will help the release of ATP. All right, so let's look at the ATP synthesis structure. Each enzyme, like I said, it has F nod and F1 subunits or parts. F nod is made out of, well, actually, let's start with F1, since that's what's on the slide. The F1 is made out of alpha beta subunits, 3 alpha and 3 beta subunits that form this propeller or turbine looking structure. It also is made out of 1 gamma subunit, shown right here, 1 epsilon subunit, and 1 delta subunit. Now, the alpha and beta subunits, like I said, form the propeller. The beta subunits have the ATP binding site. However, at any given point in time, only one of the 3 beta subunits will be associated with ATP, while another one will be associated with ADP and PI, and the third one will be empty of nucleotide. So at any given point in time, one is associated with ATP, one with ADP, and one is free of nucleotide. Alphas do not have nucleotide binding sites. They're there to support the conversion process. Now the gamma, with one of its domains that is inserted into the alpha beta propeller, that domain interacts usually with the empty subunit, but it's not just static interaction. We'll see a little bit better how actually that interaction happens. Then it forms the shaft that goes through the propeller, and at the bottom, if I can use that direction, it associates with the epsilon, which associates with the F nod side of the enzyme. The epsilon and the gamma are referred sometimes as the foot and the leg of the ATP synthase. The delta subunit connects and immobilizes the alpha, beta, and gamma structure to the F nod part of the enzyme, and so this part actually does not move. The alpha beta part does not move. So anything that has a different color than yellow except the delta is F1, and all the other subunits that are just with ABC are F nod. Now let's look at the F nod structure. 3 types of subunits, A, B, and C. 1A, 2B, again the B is used to immobilize the alpha beta propeller, and then the C subunits, we have 10 or 12 of those. They form a channel that rotates and as a result of proton movement. If the protons are going from high to low proton concentration. Here structurally in yellow, red, and orange. Here again we have the helices, and they form the channel through which protons pass through. Now let's look at the process of ATP synthesis. So like I said, at any given point in time, the beta subunits, or we can call it even in one of the neighboring alpha subunits, are in one of the 3 conformations. We have 1 beta that is ATP bound conformation, 1 that is an ADP bound conformation, and 1 that is an empty conformation. You can see even graphically they are represented differently. So let's now see how do protons play a role in the release and synthesis of ATP. So let's start with this conformation where we have now the gamma associated with this empty beta subunit. We have 1 next to it that is bound to ATP and 1 that is bound with ADP. Now 3 protons pass through the C channel. The C channel rotates, and with it rotates the epsilon and the delta subunit that is attached to it. And the delta subunit rotates from the empty and it binds to the ATP bound beta subunit. That association causes a conformational change in the beta subunit, resulting in the beta subunit changing in its conformation and losing its affinity for ATP. ATP now will be released, but the conformational change that is induced is not just localized, it's shared with the other subunits. And that conformational change now will force this ADP and PI to form ATP. And the one subunit, the beta that was empty, will now be repopulated with ADP and PI. Now, another set of 3 protons will pass through, which will move the gamma from the empty to the ATP bound, will cause conformational change, will disrupt basically its affinity for ATP. ATP will be released. The one that was bound to ADP and PI will be, the conformational change would lead to a synthesis of ATP, and the one was empty will be populated with ADP and PI. And then a third set of 3 protons will pass that will get back the conformation to where we started. So we passed 3 sets of 3 protons and we generated 3 molecules of ATP. That's a complete cycle of the ATP synthase. And this complete cycle, for this complete cycle, 9 protons were passed through the F node and 3 molecules of ATP are synthesized. So basically we need 3 protons for 1 ATP to be released from ATP synthase. This is a very important statement, and you need to remember it and not confuse it with something else I'm going to tell you later. 3 protons are required specifically for the release of ATP from the synthase. Okay. Now let's see, actually it would be interesting to see if this rotation actually really happens, and some scientists actually have done the necessary work to prove it. So it's a little bit artificial, but it proves the principle. So what they've done, they've expressed histidine tags, and all of you are familiar with those. And with those histidine tags, they immobilized the alpha-beta propeller, which normally is static anyway, but they immobilize it into a nickel slide. The part that normally rotates is the C channel. In order to visualize the C channel, they've attached a fluorescent acting filament via avidin, streptavidin, avidin biotin linker, and they can trace now the position of the acting filament. So somehow, and I'm not totally sure how they've done that, but they've stimulated the rotation of the channel, and through a camera, they detected the position of the filament, and they've noticed how the filament moved in a circular fashion. And here I've actually included a movie that demonstrates that, and you can see how this part will rotate around the central part of the molecule. This is a very similar experiment, but you can see how ATP synthase, or the channel or the F node part of the ATP synthase is rotating. Well, this one's not as fast, but this one was very nice. Okay, so to our last slide, and this is where I just didn't want you to be confused, but I'm going to make the statement that 4 protons are needed for the synthesis and transport of 1 ATP molecule outside of mitochondria. Because, I mean, indeed, yes, 3 protons are needed for the release of ATP, but that ATP is basically unusable if it remains inside the mitochondria. It has to be released in the cytosol and then in the cell in order to be used. And a transporter, a nucleotide exchanger via antiport, moves ATP outside the mitochondria, outside the mitochondrial matrix, and allows ADP to come in. The ATP synthase allows protons to come in, and that will basically capture the ADP, and with the help of inorganic phosphate will generate ATP. Well, but that inorganic phosphate also needs to be transported, which is done by a symporter that moves inorganic phosphate and a proton that's going down its concentration gradient into the across the inner mitochondrial membrane into the matrix. So you can see that for every synthesis and release, in this case, of ATP, we need 3 protons to pass through ATP synthase, but we also require 1 proton for the transport of the inorganic phosphate that will be used for the synthesis of 1 molecule of ATP. Alright, so how many ATPs are synthesized per electron pair carried by NADH, and how many per electron pair donated by succinate/FADH2? Now you've remembered up to this point, you've memorized it, that for every electron pair carried by NADH, there are 2.5 molecules of ATP synthesized, and for every electron pair carried by FADH2, given directly or through complex 2 to Q, we synthesize 1.5 molecules of ATP. Now you know why that is the case. We need 4 protons for 1 ATP molecule to be synthesized and transported outside the mitochondria. Any electron pair carried by NADH and transferred to complex 1, we have 10 protons transported across the inner mitochondrial membrane. If 4 are used for the synthesis and transport of 1 ATP, that means that for every electron pair carried from complex 1 or NADH to oxygen, we can have 2.5 molecules of ATP synthesized. 10 protons sufficient for 2.5 molecules of ATP. Now for every electron pair donated through complex 2 or directly to Q, we transport 6 protons, which are sufficient only for 1.5 molecules of ATP to be synthesized and transported outside the mitochondria. So what do you think would regulate ATP synthesis? Well, energy consumption, obviously. If you don't have sufficient ATP, that will stimulate ATP synthesis. If you have plenty of ATP, that will inhibit it. There's also another factor that you sometimes may or may not think about. But why does your breathing increase when you exercise? And usually, when I ask this question, normally I hear, well, because we need oxygen. And yes, it's been drilled into our minds that, you exercise, you need oxygen, and that's why your breathing increases. That's why your blood pressure increases, your pulse increases. Of course. And it's true. Of course it is true. But have you ever thought, why do we need more oxygen? If you haven't thought about it, well, now you know. But you have thought about it, great, because now you really have a deeper understanding of why our breathing increases when we exercise and when we perform work. And just for the sake of everybody, what do we use when we exercise or perform work? Well, a lot of muscle activity. And what's used by the muscle for muscle constriction? ATP. And you know that from 481. If we're using ATP concentration will drop. And the muscles use a lot. If ATP concentration drops, the cell will need to synthesize more ATP, Well, in order to synthesize more ATP, the ATP synthase needs to utilize that proton gradient. Well, who's going to prepare the proton gradient? Who's going to generate it? The electron transport. Well, will the electron transport occur if you don't have oxygen at the end of it? No, it won't. Ultimately, remember, all electrons will be accepted by oxygen. If you don't have oxygen, there is no electron transport, there is no proton transport, there is no ATP synthesis. That's why you breathe. Your breathing increases so that you can accept more oxygen to supply an acceptor of all the electrons that will be generated by breaking down of carbohydrates, fats, or whatever it is that is the source of your energy. All that carbon has been oxidized. All these electrons are generated, because remember, everything is about the electrons. All these electrons are generated, loaded into electron carriers, channeled to the oxidative phosphorylation system, and carried to oxygen to be converted to water. I bet many of you perhaps even now think that you breathe oxygen which will be converted to carbon dioxide. Is that the case? If you think about it carefully, you realize that's not the case. You don't convert the oxygen you breathe into carbon dioxide directly. What are you converting it to? You're converting it to water. The carbon dioxide that you exhale is a result of carbohydrates, fats, and amino acids oxidation. Remember, why are we oxidizing glucose? And what is the carbon product of glucose or fatty acids or amino acids oxidation? The carbon product is always carbon dioxide. That is a result of these carboxyl groups that are built within the molecule, and then they are released. So while the oxygen we breathe could possibly contribute to that, the oxygen we breathe is directly and for the most part used to accept electrons and to be converted to water. The carbon dioxide we exhale is a result of carbon oxidation in the form of carbohydrates, fatty acids, or amino acids. All right, that's all I wanted to say. I hope this made sense. I hope this was clear. Again, if any of you have any questions, I'll be happy to answer them. Please come and see me during my office hours, or you can email me and we can arrange to meet at a different time if the office hours are not good for you, or you can email me the question and I'll do my best to answer it. Again, thank you for your time.